Good evening, one and all. Now, we are coming to the fourth edition of the Journal Club organized by Indian Myeloma Academic Group. This Journal Club aims at actually educating all students and faculty across our country about the landmark articles and about the new aspects which are getting published every month in the field of plasma cell dyscrasias. As a part of this Journal Club, today we will be discussing the Maya trial, wherein the recent publication about the overall survival of daratumumab when given in combination with lenalidomide and dexamethasone will be discussed. Today, we have Dr. Abhinav, who is an assistant professor at Department of Internal Medicine at Armed Forces Medical College, to present the Journal Club. And thereafter, as a new addition to the Journal Club, we have with us a renowned epidemiologist, Dr. Arun Kumar Yadav, who also has a degree and a fellowship on the health economics, would be discussing about if we want to replicate this trial in our country, how would we go about it, and some biostatistical aspects about this particular trial. Thereafter, we have Group Captain Tilak, who is a known oncologist to all of us, a renowned oncologist, who will be discussing about the practical aspects of utilizing daratumumab based on the results published in this particular trial. And lastly, we have Dr. Vincent Rajkumar, who would be discussing about the nuances and the pros and cons and the real-time aspects about this particular trial in a day-to-day practice, bringing the bedside to bedside. So without delaying further, I would first request Dr. Abhinav to kindly go ahead and present the Journal Club.
A very good afternoon to everyone. At the outset, I would like to thank the organizers for providing me with this opportunity. Article that I would be covering for this Journal Club was published on October 13, 2021 in Lancet Oncology. The article reports and discusses overall survival results of the phase 3 Maya trial, which compares daratumumab in combination with lenalidomide and dexamethasone compared to lenalidomide and dexamethasone alone in a newly diagnosed multiple myeloma patient. Just covering some information about daratumumab, it is a monoclonal antibody which was granted breakthrough therapy drug status for multiple myeloma in 2013. It is an IgG monoclonal antibody which targets CD38. It has direct on-tumor and immunomodulatory action. Its progression-free survival benefit in patients of relapsed or resistant multiple myeloma was proven in phase 3 Pollux trial. And also, again, addition of daratumumab to VMP versus VMP alone showed improvement in overall survival in the ALC1 trial. Since this drug had already shown significant overall survival benefit, there is a question that why this study was designed. So this study compares daratumumab in combination with lenalidomide and dexamethasone versus lenalidomide dexamethasone alone in newly diagnosed cases of multiple myeloma who are ineligible for upfront toxic chemo and transplant. So this study setting was not explored earlier. In the SOG trial, the trial showed improvement in survival by addition of bortezomib to lenalidomide and dexamethasone versus lenalidomide and dexamethasone alone. However, in that trial, the survival benefit was found to be minimal and there was frequent neurological complications which were associated with addition of bortezomib. Coming to the methods, this trial was a multi-center trial run in 176 hospitals across 14 countries in North America, Europe, Middle East and Asia Pacific. It was a randomized open label and active control phase 3 trial. Patients included in the trial were newly diagnosed multiple myeloma patients who were more than 18 years of age with an ECOG score of 0 to 2. However, these patients were ineligible for transplant owing to advanced age or comorbidities. Exclusion criteria was extensive and the trial excluded patients with MGUS and smoldering myeloma, patients with other B-cell dyscrasias, previously treated or patients who were harboring any other malignancies were also excluded from this trial. Included patients were first stratified on the basis of ISS stage, region of their nationality, and age, and they were further randomly allocated in 1:1 ratio to the exposure group and control group. These patients received an induction and maintenance phase. The maintenance phase was in form of 28 days, every 28 days cycle, until there was any signs of progression of the disease. The primary endpoint of this study was progression-free survival, and secondary endpoints include better than or equal to complete remission, better than or equal to very good partial remission, MRD negativity, overall survival, and safety. Coming to the baseline characteristic, this slide highlights the baseline characteristics of the patient in MAIA trial, median age being 73 years. I would like to bring to the notice that 92% of the patients included in the trial were whites. Thus, generalizability of results for minority groups may not be accurate. Similarly, when we see 84% of the patient had a performance status of 0 to 1 on ECOG scale, which may not be true in the real world scenario. In this slide, we see that randomization was done post stratification as per the age, ECOG score, and ISS stage. And after stratification, these patients were randomly allocated in a 1:1 ratio in DRD and RD groups. Looking at the outcomes, a total of 737 patients were randomly assigned into these two groups. In DARA group, 364 patients received treatment, while in RD group, 365 patients received treatment. We observe a higher discontinuation rate in the RD group owing to a PFS event or an adverse effect on or a physician decision to switch the treatment. We observe a higher number of patients remained on treatment in DARA group. Coming to statistical analysis, I would like to bring to the notice that the sample size calculation by the in this study was by the PFS assumption alone and was not based on overall survival assumption. So interpretation of the overall survival in this update may not be accurate, as the study was not powered for overall survival. In the update, at a median follow-up of 56.2 months, out of 377 patients who had a PFS event, 43% of the patients belong to the DARA group and 59% of the patients belong to a control group. Thus, it establishes the PFS benefit in the DARA group. Out of the 160 events which occurred in the DARA group, 30% were death of the patients and 70% were disease progression event. While in control group, 217 events occurred, out of which 21% were associated with death of the patient and 79% were disease progression event. The analysis thus shows overall survival benefit in the DARA group. As we can see, forest plot of the study in almost all groups comparing the overall survival favored the DARA group. To summarize, at 56 months of follow-up, statistically significant improvement in overall survival was reported in DARA group, although sample size was not based on overall survival assumption. Significant progression-free survival benefit was reported in primary analysis, was maintained in the current analysis also. A higher proportion of patients achieved the complete remission or better in the DARA group. Also, the patient in DARA group fared better on next line of the therapy. This is a table showing comparison of primary and secondary endpoints. It shows in the current analysis that almost all endpoints favored the DARA group. There were no new safety concerns which was identified with the longer follow-up. Most common adverse events being neutropenia, anemia, pneumonia, and lymphopenia. Grade 3 or higher infections were reported more frequently in the DARA group. This table shows other adverse effects associated with the treatment and their frequency in both the groups. Now, coming to the critical appraisal of this study, the PCOT analysis. A total of 737 participants were included in the trial. 368 patients were allocated in the exposure group and 369 patients were allocated in the comparison group. Duration of the study was between March 2015 to October 2021, and the outcome showed significant progression-free survival and overall survival benefit in the exposure group. Assessing the validity of this study, we have used the RAMBO acronym for assessing the study bias. R standing for recruitment, A for allocation, M for maintenance, M for measurement of outcome, and B for blind and O for objective. Coming to the recruitment, study setting and eligibility criteria were well defined. However, representativeness of the participant remains questionable, as despite being a multi-center trial, the included population mostly comprised of whites, 92% to be exact. Again, the prognostic and risk profile were not representative of the real world scenarios. The study favored patients with good performance status. The randomization process was described adequately in the study. Allocation to the both groups was done after stratification as per age, ISS stage, and nationality. And further randomization was done, and the study group was divided into 1:1 ratio to both exposure group and comparison group. Most participants did remain allocated to their respective groups. However, the physician and patient had the option to switch to the exposure group. The trial was an open label trial, so the participants and investigators were not blind to exposure. Compliance was found to be more in favor of DARA group. The follow-up was high and similar in both the groups. Measurement of outcomes were objective and definitive and was laid down in the protocol, and the authors did review all the results. Coming to the title, it was interesting. The abstract conclusion will be useful in clinical practice. However, setting of the study was not representative of the general population. There was clear research question. The authors had appropriate facilities and technical expertise to carry out this study, and there was a novelty to the study. However, a selection bias was possible as it was a open label study. Coming to the limitation of the study, an open label study design probably led to a bias for early patient withdrawals from the RD group. Pre-specified subgroup analysis of overall survival includes certain subgroups with a very small sample size. For example, like non-white population or patients with impaired baseline hepatic function or renal function, or patient with high-risk cytogenetics. Further, the primary endpoint of the study was progression-free survival rather than overall survival. This endpoint was suboptimal to address the clinical question because previously daratumumab has already shown a progression-free survival benefit in RRMM cases. Also, the current study, though, was reporting overall survival, but it was never the primary research question for Maya trial. The sample size calculation was again never based on overall survival. The trial was also used a controlled treatment that is no longer the standard of care. The addition of bortezomib to lenalidomide and dexamethasone has proven survival benefits in the previous trials. Also, the study arm received additional drug in induction as well as maintenance phase. Thereby, it maximizes any possible benefit in the surrogate endpoints of progression-free survival, complete remission, and MRD negativity. To conclude, taken together, the study data showed longer overall survival with DARA RD versus RD alone in transplant ineligible newly diagnosed multiple myeloma cases. The study recommended early addition of DARA for the treatment of transplant ineligible patients in newly diagnosed multiple myeloma patients. Both these study conclusions are fraught with multiple caveats, as has been discussed above. Thank you.
Thank you, Dr. Abhinav. It was a wonderful presentation. So now coming to the, I would now request for people who have joined late, just a quick introduction about Dr. Arun Kumar Yadav. He's an epidemiologist. He is MD, DNB, and PhD from AIIMS, New Delhi. He is MA in sociology. He is a MAMS, FIPHA, and FAIPI. He was ex-professor of community medicine at Armed Forces Medical College. He is a joint secretary of IPHA West, ex-governing council member of IAPSM, ex-editorial board member of IJCM, and he is a member secretary for IEC, lead nodal officer for the multidisciplinary research unit at AFMC previously. And he is also DHR for ICMR. So now I would request Dr. Arun Kumar Yadav to kindly comment about the biostatistical aspects and how to replicate this study in our country. Over to Dr. Arun, please.
So, thank you, Dr. Uday. Am I audible?
Yeah, you are. Please go ahead.
Okay, thank you so much. Can I have my slides or I show it from my end?
You can also share if you want, sir.
Okay. So I am sharing my screen. Okay, so is full screen visible?
Yes, sir.
Okay. So good evening, everyone, and thank you, Dr. Uday, for giving me this opportunity to be here with you all and give out my thoughts over this study. So as very well brought out by Dr. Abhinav, so I will start with conflict of interest. Though the author has brought out in one para what is their conflict of interest, but in this, please remember, sponsors of the trial had got complete access to the data. They were not only in access of the data, but they are part of the writing team. So that is something which should be seen with a little skepticism. So, however, the point over there is that they have declared it in their paper. The other thing is that doing a RCT over 176 trial units with among 14 countries is commendable. However, when you go to their site, which is there where they have registered over there on NCT site, and when you once register, you have to give a certain document, and this document includes their protocols. So once you go to their protocol, there's the site registered at 256, and none of the sites, I repeat, none of the sites which is registered for the trial is from Asia Pacific or African regions. So I don't know whether they have changed it later on to 176 and included few from outside America and European. However, in this paper and the paper previously which they have published, they have said that they have sites from outside US and Europe. Now, coming to their statistical plan, since it's a study which have got time to event, so either it is progression-free survival or overall survival. So the basics are the data should be presented in person years or person months. It is never presented so that when you have hazard ratio, you have simultaneously person years or person months. It is never presented as percentage. So I was looking whether authors at any point of time in their complete paper have presented person months somewhere. So they have presented it only once for the secondary malignancy, because in case of secondary malignancy, if you see the chart which has been shown by Dr. Abhinav, that secondary malignancy was much more in case of DARA arm. And later on, when they calculated person months, it came out that it was 4 per 100 person months and 3 per 100 person month, and they concluded that it was not a difference. So similar things can happen with all the percentages. So if I would have been there, I would have presented it as a simple case of person months or person years. Then in sample size calculations, so they have taken for estimation initially for calculation of sample size, they have taken at 0.75, hazard ratio of 0.75 for calculation, for which they have come out with 390 events. Now I tried to search, I don't know for what reason they have not given any reason or the reason for assuming this 0.75 hazard ratio for calculation of sample size. Other thing is that whenever we calculate sample size in multicentric study, please remember a patient in America has got more probability of going to an American clinic rather than coming to European or Asian clinic. So there has to have some kind of cluster effect. So whenever we calculate sample size for any multicentric trial, and it has, unless and until there is something to assume that yes, all patients are independent, we generally feel that the patients which are coming to one center might have some kind of peculiarity which is to that center. So no cluster effect was taken. Another peculiar thing with this is that they have done randomization stratification at the time of randomization, and then they have written in their when they have done in methodology, they have written that they have taken stratified Cox proportion. I was trying to find out in their paper where is that diagram of stratification Cox proportion, which is not there, because these two things do the same thing. They reduce the effect of the variable for which stratification has been done. Now, if you stratify, they have stratified for three, in randomization, they have stratified for three factors. One has got three groups, other has got age, has got two, and another one variable has got two. So there are total 12 groups out of 700. It says that hardly they have 58% to make two groups into it. And another thing is that if you do randomization with 58% only, your few variables, confounding variables, will not be adjusted. However, when they have presented the data, it seems that there is a perfect randomization, which is, to me, which looks little skeptical. In analysis, if you see progression survival curve, which has been shown, progression survival curve is a copy book curve. I mean, there cannot be nothing classical curve for a reduction of hazard than the progression-free survival, which has been shown again. Getting such a curve in real world is, to me, unless and until I see the data, I am little skeptical. Now, a lot of things have been spoken about generalizability. Of course, for generalizability to our settings, especially low middle income settings, the participation, their population has already been brought out by Dr. Abhinav, is different. So generalizability to our population is little doubtful. Other thing is that in their study, since it's an open label, sponsor driven, many centers are involved. Please remember, when there are many centers involved in any study, then the chances of dilution of your standardization, of your training, of your inclusion of patient to the study gets diluted. And it has been shown in the study itself, wherein two patients, which ideally should not have been part of the trial, because of their inclusion and exclusion criteria, have become part of the trial. And the authors themselves have mentioned that ideally they should not have been part of the trial. Other thing is, if there are many centers involved, your protocols get changed, your treatment in between gets changed, which they also admit that sometimes they have to stop one drug, two drug, and continue another drug. So all these variables are there. With this, generalizability becomes limited. Now, if we have to do this study based on, if we have to do this study in our low middle income country based on the study which we are seeing. See, in this study, the hazard ratio for progression-free survival is 0.52 or 0.53. So for doing a study, you require only 78 events. That means that you require 150 patients only in one arm. So, for if their study is correct, suppose for a so sample size straight away reduces to nearly sample size reduces to nearly 1/3 to one or nearly about 1/3 of their original sample size. Now, during COVID, and so there are proposals of doing adaptive trials, and to have some benefit to low middle income country, all these trials can be piggybacked with health economic study, which is generally a norm in all the RCTs these days. Adaptive trial is best when there are multiple treatments, multiple options are available beyond standard of care. Now in this, as has been brought out by Dr. Abhinav, that standard of care has changed and there are multiple options available. So with the same number of patients, we can have adaptive trials, can have faster results. And I think adaptive trials will be best suited to be doing in LMIC countries, wherein we can piggyback it with our health economic study to come out the cost of each new regimen which we are providing in these countries. And of course, then our population and participant, one of the things in population and participant is the age. So their median age, if you see, is around 73. And in our, this thing, if you see, our life expectancy is still around 71. So we do not expect many patients as such to be living at to such age. So we have to have good participation from our kind of patients. Again, sample size I had spoken about, that sample size needs to be recalculated. With this study, the sample size comes out around 78 event points. And again, sample size reduces if this we take findings of this study. In randomization, I would rather do randomization not with all these stratification, one-odd stratification, and then I would like to adjust it with Cox stratification. Besides this, the study which we should design should have important results which can help clinicians in real life settings. So if clinicians feel that overall survival is more important, we should design studies with overall survival. Similarly, if health economics in low middle income countries is important, we should have health economics built into our study. Now, even if we prove, please remember there are 2-3 terms, that even if we prove efficacy in our trial, we have to go 2 steps further. One is effectiveness, that will be when our patients coming to us. Another is there might be some problem of implementation. So we have to rather cross these 2 steps further of effectiveness and implementation, even after proving efficacy of the drug. So that's all from my side. I'm ready to take on any questions if there are any. Thank you so much. And thank you once again to the organizers for having me over here. Thank you.
Thank you, Dr. Arun. It was a thoughtful insight about the statistical aspects of a trial. And I have liked the way you ripped apart the trial, saying that there are so many lacunas. But still, I think now we will listen to, firstly to Tilak sir. The audience who are here are not new to this name, Group Captain Tilak. One of the most loved and the favorite hemato-oncologist or oncologist in the armed forces circles, and he is an alumnus. He did his MBBS and MD from FMC and DM from AIMS. He is a known figure as far as the country and the oncology circles are concerned. I would request Tilak sir, based on the inputs which are given the presentation by Dr. Abhinav and the biostatistical inputs by Arun, sir, how would you take up the results of this study for a day-to-day care in our country?
Thank you, Colonel Uday. At the outset, my deep sense of gratitude to the Indian Myeloma Academic Group for giving me this opportunity in this Journal Club discussion. and especially to Colonel Uday for having me on board. It is indeed an honor and a privilege to be amongst the stars of myeloma, both internationally. I can see Dr. Vincent Rajkumar and those from the country. Thank you very much. Secondly, thank you, Dr. Abhinav, for the nice presentation of the journal, and Colonel Arun for the insightful comments. So this particular study already discussed in detail. So there are some goods and there are some not so goods, we can take it that way. So what are the goods which has happened? One is that a new drug which has been included into the transplant ineligible patients pro forma has been included, and like my dear friend Colonel Rajan Kapoor calls it, Dara Singh has been included into the protocols. So this particular drug has got an advantage of using subcutaneous in addition to the intravenous. So that is one good thing. It can be it can reduce the decay time, etc., and it can add a new protocol in myeloma patients. Secondly, more than a large percentage of patients are beyond 75 years of age. So the elderly population have been included into the study who are classically transplant ineligible, and it gives a new kind of a regime for these kind of patients. Thirdly, it does have progression-free survival benefit as shown in the primary analysis, and the subsequent paper which we discussed today showing the overall survival benefit. What are about the not so, I wouldn't call it not so good, but some limitations in the entire study. 33% of the patients had a PS of 0, which means 1/3 of the patients had actually nearly no symptoms. Is it that these people were having a good disease biology and would have probably done better anyways? And how much did DARA add to that particular effect is one thing which needs a review. Secondly, overall survival not being the primary endpoint in the initial trial that have all been discussed in details. Secondly, the endpoints of the trial in our country, if you want to use daratumumab, we would rather go in for a trial that's got a definitive overall survival benefit. Reasons being, one, you're trying to change practice here. Number two, the cost of the drug is high, and especially in resource limited settings, PFS probably would not be an ideal endpoint for a practice changing kind of a paper. So from that point of view, this paper has slight limitations. There was reported incidence of more deaths in the daratumumab group, especially those who discontinued the treatment. So that leads a little bit more into decision as to why did these patients who were on daratumumab have more deaths as compared to the one in the lenalidomide group. Secondly, the duration of treatment of use of daratumumab. They have used it in the induction, the consolidation, the maintenance. So the actual duration of use of daratumumab is still not very clear. Is it some way we can probably choose patients in our country, a subset of them to whom we need to offer daratumumab, and do we need to give them throughout the entire treatment course, or is there some subset where a course of treatment where we can have daratumumab included? For example, the phase 3 Cassiopeia trial, which used DARA plus VTD versus VTD alone. In these patients who had DARA plus VTD in the induction consolidation followed by the maintenance, there the PFS advantages were not seen very much as compared to the VTD patients who got DARA only in the maintenance phase. So can we use something like this kind of a setting where the cost of the medicine can also be adjusted and the patient also gets the benefit, especially in low and middle income countries and resource limited settings? Of course, the advantage of subcutaneous has already been mentioned. The dose of lenalidomide reduction was there in those patients who required daratumumab, the lenalidomide dexamethasone arm. So yes, probably the reduction of dose might lead to a reduction of the secondary malignancies. However, the cumulative dose of lenalidomide was much higher than in the plain lenalidomide dexamethasone group. So in the Indian setting, elderly patients, transplant ineligible, in the debate of 2 versus 3 drugs, if we have to use 3 drugs, we have a winner already with bortezomib. Daratumumab is inching closer, but probably my take would be that we need a little bit more data with a stronger overall survival analysis to use it with probably a limited kind of a duration rather than using it to the entire duration of the therapy. These are a few comments from my side. One last point could be that in the immuno-oncology drugs which we use in solid tumor oncology, we have started using the low dose therapies, where we have come down the dose almost 1/3 the recommended dose, and we are finding beneficial effects. Is there some way we can reduce the dose for daratumumab for its effect? Needs again more of a speculation, but can be studied. With this, I hand it over back to the organizers.
Thank you, Tilak sir. Now, we have the final comments from the who's who of myeloma, Dr. Vincent Rajkumar sir. Sir, your comments, please, about everything, starting from the article to the comments to the biostatistician to the Indian Vala perspective, everything. Thank you, sir. Over to you.
Thank you so much again for having me. And I'm really, really sorry that last time I missed the VRD versus KRD discussion. So I might throw in a couple lines about that as well. I mean, really, first of all, amazing discussion. I don't usually get to be in journal clubs where people have read the paper in such great detail and have really analyzed all aspects of the study, like I heard from Dr. Arun, Dr. Abhinav, and Captain Tilak. It's also important that the final conclusion that Dr. Tilak made is like exactly the same conclusion I have said even for the US, which is we already have a great regimen, VRD, and even in a country like the US where both regimens are available and covered by insurance, generally I would still prefer VRD, primarily because of the dose and the schedule and the cost, and that I would need more data on overall survival benefit of DRD versus VRD to make a switch. And even if I were to switch, I would rather do a fixed duration rather than a prolonged indefinite therapy of DRD. So the conclusion that you gave is just the same conclusion I have reached. Having said that, in a journal club, and when I hear these criticisms and all of the points that you made, I think some things are important to keep in mind because they don't come out in any written paper, and you have to know behind the scenes or basically what is going on to get an idea of why the trial was done the way it was done. So a few years ago, Dr. Moreau and I wrote a paper in which we said that no matter how many times you read the protocol, you never get an idea. It's never written in the protocol, why was this study done? Why was the study done is very different from what is the primary objective of the trial that is written in the protocol. Primary objective of the trial written in the protocol might simply say, we want to find out whether DRD has better PFS than RD. The secondary objective might be we want to find out whether DRD has better overall survival than RD, whether the response rate is better than RD. But that's not the same as why was this trial done, which is a different reason. And that reason may be known to the person who does the trial, who pays for the trial, but not the people who are enrolling, may not be apparent to the people who are reading the paper or even having to use the drug. And that then leads to a criticism of a trial from one angle, which doesn't understand why was the trial done, which, as I said, is not revealed. And I basically wanted that to be added to all the journal articles so that the readers know the answer to that question, in which case then they will be able to interpret these kind of studies better. So why was this trial done? This trial was simply done to secure approval in the frontline setting in the US and Europe. It is not done to find out what is the best treatment for newly diagnosed myeloma. It is not done to find out whether VRD is better or DRD is better. It's merely to make sure that this drug daratumumab, which was shown to be effective in the Pollux trial in the 1 to 3 prior relapses, can get a new indication in the newly diagnosed setting so that the drug can be sold and reimbursed in the US and in Europe. And if if I, as a physician in the US, or you are as a physician in India, want to find out what is the best treatment for newly diagnosed myeloma, DARA RD or VRD, we have to find the money to do the trial. We have to find the drugs to do the trial and answer the question. It is not really, from a drug company perspective, their responsibility, in the sense that they are spending hundreds of millions of dollars and they want to find out an answer to the question that is important to them. And that's where the investigator-initiated trials have to be the ones that answer the kind of questions we want to see answered, which is DRD really better than VRD? If I'm going to give DRD, should I really be giving it forever, or should I just give it for like 2 or 3 years and that will be enough? Those are questions that might be great medical questions, but that's not the immediate pressing question for pharma to answer. And they're also not the immediate pressing question where they will spend the money, because they are the ones with the skin in the game. If they lose the trial, they lose the hundreds of millions of dollars. You and I would not lose any money out of this, and we would just carry on with our lives that we didn't find the answer. So keep that in mind. Now, I did write a big criticism of the Maya trial when it came out, and it's in Nature Reviews, and we wrote it with Dr. Prashant Kapoor, stating many of the same questions that, even if this is the reason, you as a user, we have the right then to interpret the study the way we want to interpret it. You can do the trial whichever way you want, you can get your approval, but we'll interpret it the way we want. And I think that's what all of the criticisms and the final conclusion led us to. I will tell you that there are some important differences between the US and Europe and why this trial was designed the way it was designed. In the US, progression-free survival benefit alone might be enough for the FDA to approve a drug. And if that's the case, the company is going to try and get the approval for PFS. And they know they can get the PFS answer several years before the overall survival answer, which means the drug is on the market for newly diagnosed myeloma and can be reimbursed years before if they were waiting for overall survival. In Europe, many, many times, and the companies will never know the answer for sure, but many, many times regulators want to see the overall survival benefit. And without the overall survival benefit, they might not give approval. And so many times when they're designing the trial, when you look at the sample size, it won't be entirely obvious. After all, Pollux had a sample size of 0.3 something. They could have made the study 1/6 the size if they wanted, or 1/4 the size. But they're not going for that. They're spending hundreds of millions of dollars, and therefore they want to make sure it's safe. They want to make sure that they will get an answer on the overall survival in case the European regulators want that answer. They want to make sure that the PFS result is they don't do a trial for 5 years and find out that the PFS result is not there. They have the money, they're willing to spend it. Both regimens are reasonable for newly diagnosed patients. The physicians don't feel an ethical problem giving either regimen, RD, and then patients can get DARA, which is approved for relapse, or start with DRD from the beginning. So you see the sample size is higher than what you think you need, but that's because of that particular reason, which is there is a sponsor trying to do a study, but they are facing a regulatory outcome which is different in US and Europe, and they cannot say they want to do the study with the primary endpoint of overall survival, then the FDA won't act on the PFS result, but they have to have the power for the overall survival, otherwise the whole study will not get any label in Europe. And so they end up having some trial design where the sample size calculation doesn't make sense to a statistician who's looking from afar, and they say, well, why did they power it? Why did they overpower this trial? And why did they have this endpoint? And even for the reader, you were wondering, why didn't they use overall survival initially? Well, that's because if they can buy and sell the drug, they can sell the drug and get it reimbursed with PFS because the FDA is willing to go with that endpoint. Why would they wait and do the overall survival? So I hope that answered the question, and it raises the point that why the trial was done is not written in the paper, and it's not part of the thing. Then you go into the number of centers that they enroll and the question of race and things like that. So what happens is they have the money to do the trial as fast as possible. And so they will open it in as many sites as possible because they want the result tomorrow. Every day is like several $1 million in loss if they don't get the drug approved. So they're always trying to do these trials as fast as they can, open it in as many centers as they can. When they do that, what happens usually is that two reasons. One is in USA, trials are slow to accrue, slow to open, slow to accrue. In Europe, and part of the reason why they're slow to accrue in the US is that we could give DARA sometimes for well-insured patients outside the trial, even before it's approved for frontline. So physicians who feel like we don't want to give a doublet and we don't want to give VRD, they can give the DRD anyway. So by the time US centers like Mayo Clinic and all start opening the trial is already accrued in Europe, and we are hardly able to keep it open for a few months when the trial meets its accrual goal, which means that the trials, all the randomized trials, not just this, any company-sponsored randomized trial will have very few US patients, and as a result, very few African Americans or Black patients. And that tells you why the racial disparities are so huge in phase 3 trials. The control arm is always a problem because when the trial is done, we all knew in India and in the US that VRD is what we would use. And in fact, India is one of the few countries besides the US which for many years could use VRD because both bortezomib and len were cheap. In the US, both bortezomib and len were reimbursed. So we were using VRD before all other countries, India and USA. In other countries, people knew that VRD was better, but even in Europe, it was not approved for frontline. And so you have a trial like Maya designed where we are all using VRD. We all think VRD is the best treatment. And if at all somebody should do a randomized trial, it should be DRD versus VRD. But the reality is that the control arm that is being used ethically in all these other countries is RD. And as a sponsor, they go like, they have the same calculation. If I use VRD versus DRD, I need twice the sample size, longer time to show the benefit. Why would I do that trial when I can get the same result faster, quicker, less patients with an RD control arm, which ethically is okay in all the countries that I'm trying to do the trial. And so then they end up with a control arm like this. Regarding conflicts of interest are, I usually say no conflicts of interest, but I want to make sure that you understand I am an absolute exception to the rule. A lot of people have conflicts of interest, like Dr. Fakon and others, mainly because they are leading so many trials. And when you're leading a lot of trials, phase 1, 2, 3, you end up being on steering committees and data safety monitoring committees and everything. And time is money, money is time. So they are reimbursed for work that they actually do, which then shows up as conflicts of interest. I'm getting away with having no conflicts of interest only because I have tried not to lead these trials at Mayo, which I'm able to do since I have other investigators who can lead the trial, and I already led enough trials. And therefore, I'm able to keep myself free of conflicts of interest. But it's not really practical for anybody except people who never do clinical trials to say that they don't have conflicts of interest. So you will have those conflicts of interest. Regarding the trial itself and the 2 patients who got left out and what to do and how they should have done it, these regulatory trials compared to trials you and I do are monitored. Monitoring means every patient enrolled, every patient had any lab done is actually seen and checked within a month or 2 of that event happening by an outside agency. And they keep on monitoring this trial because when they submit these data to the regulators also come and audit, and they don't want any mistakes to be found. So the chances that there can be any errors or problems or anything is far fewer in a company-sponsored trial than in a trial that investigators would do ourselves. Because in investigator-initiated trials, you do audits. You occasionally will check an institution to see whether everything was correct. But in a company-sponsored regulatory trial, it's actively monitored. So they catch mistakes. Like there might be the occasional patient who got through, but within a month or two, the monitoring team would say, you enrolled this patient, this particular reason the patient was not eligible. Why on earth did you enroll the patient? The center is cited. And so everybody is monitored very actively. So the chances of mistakes, errors are all extraordinarily low. And you and I could not do a trial like this with this few mistakes. As you know, even if you were monitoring our trials, you'll find a lot more problems because it's impossible to not make mistakes. And they tend to have far fewer because they know the risks that are there. One other point that was brought up was, and I'm trying to remember here, low middle income countries and health economic costs. You know, India has always been a problem for many countries, particularly Celgene, BMS, Takeda, because we have multiple generics for the same drugs that they have. These countries also realize that DARA cannot be quickly made into a generic like lenalidomide or bortezomib could. Then they make the calculation of where exactly am I actually going to make money selling these drugs, and those are the countries that they will open the trial in, because their interests are different than your interest or my interest. The drug they've already developed, they've already gotten the drug approved, it's on the market. Now they're trying to get a frontline indication. It costs a huge amount of money to do these trials. And so they end up doing this. So I think we have to look at it that way and then go to see what can we do now that this has happened. As you know, India was the only country where pomalidomide has been studied in a frontline setting in multiple trials now. And that's because only you can do the trial. If I want to do a trial with pomalidomide, I will have to pay for the drug because the companies won't even provide the drug, let alone the funding. So if that's the reality, and therefore countries like India, you may want to do the exact IIT that will answer the question that is helpful to you. And that was easy with pomalidomide and bortezomib and len because they are inexpensive. But with DARA being so expensive, I'm not still sure whether you can pull off a VRD versus DRD trial. But if there ever was a VRD versus DRD trial that will happen, it'll only be possible in India. And so you have to keep that in mind. And if that endpoint is of interest to you, that's something only you can get away with. Similarly, if there was ever a trial where you ask the question whether 2 years of DRD is as good as indefinite, probably only India could do that trial because, again, you could do it as an IIT there with commercial drug, whereas in the US and Europe, it won't be possible. And then the open label question. See, DARA is impossible to blind. Anybody getting DARA would know immediately that they're getting DARA, basically based on the side effects that happen with the first dose. So a placebo-controlled trial is just not possible with DARA because you would be only doing a fake trial, since everyone would know just based on the cycle 1, day 1 dose, who's getting DARA and who's not getting DARA. So that's why they don't play these games. They basically accept the question that this is not possible to blind these drugs, and therefore let's do a unblinded trial. There is a trial that was done with blinding because blinding was possible. That's ixazomib. And you know what happened. Yes, the progression-free survival benefit narrows because patients, it's truly double blind and you don't know who's getting what. And then the patients who drop out are uniform. Whereas you see what happened in this trial, as Dr. Tilak pointed out, I think that, or Dr. Abhinav, that treatment, adverse events that led to treatment deaths, he was wondering why they were slightly higher in the DARA arm. They are slightly higher, but that I don't think that's that important. It might be just a chance effect. Because there's like 2 or 3 numbers the deaths due to progression, of course, the overall deaths are much higher with with the Lendex arm than the Dara Lendex. But the important thing is who discontinued treatment and deaths that happen in those patients due to adverse event. I think the number is 40 versus 82, if I'm not mistaken. And that's really important because, so you can see here, treatment emergent adverse events leading to treatment discontinuation were reported in 46 patients in the DARA group and 82 in the control. And that's purely because somebody's on DARA does not want to leave the DARA trial, because if they do, they'll never get the DARA again. Whereas in the control arm, they're very willing to stop the trial because both drugs are available commercially, can be added with other drugs and used. So these things happen, and I think the criticisms are valid, but there are reasons why some of this is inevitable, and you have to keep that in mind, and you have to really go into who's doing the trial, what are their interests, why should they care about what's interesting to you or me? And then if we are interested in a question, we have to do it. And that's why the VRD versus KRD trial was done the way it was done, because for Dr. Kumar and me, the question was not whether KRD is better than RD. The question was really whether KRD is better than VRD. The companies are not going to do that trial. So we did it as an ECOG trial. When we do it as an ECOG trial, it accrues very slowly and it takes a very long time to accrue. And by the time the results come out, you know, we have already moved on to DARA RD, so the KRD question is not as important. But we did find the right answer, which is that no matter what the phase 2 trial showed, KRD was just as good as VRD, VRD was just as good as KRD, and we were able to keep a second randomization with the survival. And if you see, that trial is also overpowered because we are trying to answer an OS question for the second portion of the trial. So in summary, the Maya trial has drawbacks, but it was done with an important goal in mind, which is to provide access to DARA in the frontline setting for the US and European markets, which it did successfully. From a clinical perspective, that doesn't mean we have to use DARA RD. We know that it's an option that's available to patients, and then we have to decide when to use DARA RD, when to use VRD. I look at the trial data for the people over the age of 65, because the VRD trial, the SWOG 0777 included patients of all age groups. But if you look at over the age of 65, the 5-year survival rate in the Maya trial was 66% for the DRD arm and 55% for the VRD arm. Now, I don't think that difference means that DRD is better than VRD for overall survival. It's hard to compare trials because Maya was done about 3 years after the SWOG trial finished its accrual. So the salvage options are more for patients enrolling on Maya, and that could explain the survival difference just in that way alone. The median overall survival was more than 60 months for VRD and more than 65 months for DRD, more than 65 months for VRD and more than 60 months for DRD, and that is for people over the age of 65. So both regimens are equally good from an efficacy standpoint. I prefer VRD still because I can use the triplet for 6 months and then do just lenalidomide maintenance. Whereas with DRD, to get these kind of results, I would have to use the full triplet until progression. So that's more expensive, more toxicity. My colleagues tell me in the US that even if they use DRD, they don't use the Maya schedule. They give the DARA RD maybe for a year or two and they stop it, and then they'll just give the lenalidomide as maintenance. So for those of you using it in India, you could stop it after a year and then just do len for maintenance as an alternative. So I'll stop with that. Great discussion. Let's have some questions.
Thank you, sir. Sir, it was phenomenal discussion. Among all the journal clubs till date, I think this was the best discussion ever we had in the last few journal clubs. I Actually, because of the paucity of the time, I would like to, because we have got two more events to go. One is the general scan and the quiz to follow. So, and there are no major questions in the chat box because those have been answered. And for Dilip Mevada, who has requested for ASH highlights, I would request you to participate in the upcoming Indian Myeloma Congress at Bangalore in St. John's, where all these panelists and all these international speakers are going to tell you about the latest as far as the ASH 2022 is concerned. Now, coming back to the general scan, so we have this time forwarded the Castor trial, the updated results for the general scan, and we have the commentaries from different people. And the best commentary that we have had is from Dr. Mona Vijayan from Lucknow Sahara Hospital. So, Mr. Nirav, can you just play the general scan from Dr. Mona Vijayan?
Good evening, everyone. I am Dr. Mona Vijayan. Today, I will be presenting the journal scan for this month. The topic of the journal scan is overall survival with daratumumab, bortezomib, and dexamethasone in previously treated multiple myeloma patient. It is the Castor study. It is a randomized open-label phase 3 trial which was published in Journal of Clinical Oncology in November 2022. The purpose of the study is that at the primary analysis of Castor, which was done at 7.4 months, when DVD versus VD was studied, there was a prolonged progression-free survival in relapsed refractory multiple myeloma patient. The Castor study now updated efficacy and safety results at the final analysis. So daratumumab is a human IgG-kappa monoclonal antibody targeting CD38 with a direct on-tumor and immunomodulatory mechanism of action, demonstrating greater cytotoxicity towards multiple myeloma cells ex vivo compared with analogs of other CD38 antibodies. The Castor study now has done a 6 year of follow-up and now provides the final overall survival analysis. Methods. As said, it is a phase 3 study in relapsed refractory multiple myeloma patients. The eligible patients were who had received at least one prior line of therapy, who had at least a partial response or better to one previous line of therapy, and the patients who were refractory to bortezomib or another proteasome inhibitors were ineligible. Random assignment and study treatment protocol. In the VD group, they received bortezomib in 21-day cycles on days 1, 4, 8 and 11 and dexamethasone 20 mg orally on the following days. And daratumumab group, the daratumumab was given at a dose of 16 mg per kg once weekly in cycles 1 to 3 and once every 3 weeks in cycles 4 to 8 and once every 4 weeks thereafter. The endpoints were PFS was the primary efficacy endpoint, secondary endpoints were disease progression, overall response rates, MRD negativity and overall survival. The results were 597 patients were assessed for eligibility and 99 were excluded, and randomly assigned were 498 patients. 251 were allocated to daratumumab group and 247 were allocated to control group. This is the Kaplan-Meier estimates of overall survival. There was an 11-month gain in overall survival if daratumumab is added to bortezomib and dexamethasone. The median overall survival was 49.6 months for DVD and 38.5 months for VD. The Kaplan-Meier estimates also shows a progression-free survival in the intention to treat population with DVD. Here is a Kaplan-Meier estimate of overall survival by MRD status. It is a very important slide. It shows that there is a superiority in overall survival when is derived in patients who achieved MRD negativity in DVD group. But the overall survival was similar in DVD and VD groups who were MRD positive. Now this is the forest plot which shows that if the patient had received only one prior line of therapy, then the response with DVD was far better than if the patient had already received 3 or more lines of therapy. Safety. In the patient who were on DVD group, they had higher thrombocytopenia, neutropenia and lymphopenia. Now, it is the Castor study is the first overall survival data with daratumumab in the relapsed refractory multiple myeloma population. There is a 26% reduction in the risk of death at a median follow-up of 72.6 months. The grade 3-4 infections were more frequent in DVD and second primary malignancies were higher in DVD. The weaknesses of this study is that now the triplet has been analyzed with the doublet, and the strength of the study is that it supports early use of daratumumab to induce deep and sustained responses and prolong disease control to potentially delay clonal evolution and subsequent drug resistance. Thank you.
Thank you. Thank you, Dr. Mona Vijayan. Mona Vijayan is she's a clinical hematologist who has done her DM from AIIMS, New Delhi, and currently is practicing in Sahara, Lucknow, Sahara Hospital at Lucknow. So next, can I have the quiz questions, please, Mr. Nirav?
Just a
So we have, as usual, as part of the Every Journal Club, we have a quiz which constitutes of around 5 questions. And these questions you have to answer in 30 seconds time, and the time starts from the time the question is flashed. And we'll be explaining the answers later. Now, most of the questions are so easy to answer that if you would have concentrated in the last half an hour, 45 minutes about what was being discussed, you can actually answer most of them. And the winner gets an Amazon voucher worth 1,000 rupees. You have to select one out of the 5 options which have been flashed here. There is no negative marking. So we want all of you to give it a try. Okay, can we have the first question, please? Institute used daratumumab for the first time in India among the options shown here. AIIMS, New Delhi, PGIMER, Chandigarh, Christian Medical College, Vellore, Army Hospital, RNR at Delhi Cant, none of them. In fact, this institute has also used the daratumumab for the amyloidosis indication for the first time in India. This we are talking about in non-research or non-trial scenario. Okay, coming to the second question. All of the following trials are related to daratumumab except Pollux trial, Castor trial, Maya trial, Cassiopeia trial, none of the above. So which of these trials is not a Dara-based trial? The third question. All are the labeled indications for daratumumab in newly diagnosed multiple myeloma patients, except in combination with VTD for transplant eligible patients, in combination with VMP for transplant ineligible patients, in combination with Lendex for transplant ineligible patients, in combination with Botidex for transplant ineligible patients, or none of the above. We are talking of newly diagnosed multiple myeloma patients, not RRMM. Fourth question, the correct prescribed dose of daratumumab when you are giving it in combination with Lendex in newly diagnosed patients of multiple myeloma, it's not NDDM, it's NDMM patients, is 16 milligram per kg once weekly for 6 doses, followed by once in 3 weeks for 16 doses, 16 milligram per kg once weekly for 8 doses, followed by once in 2 weeks for 8 doses. For 6 doses initially, once weekly, followed by once in 2 weeks for 4 doses, once weekly for 8 doses, followed by once in 3 weeks for 8 doses. Please understand, Dara combination with different combinations are different dosings. We are talking of in combination with Lendex. The fifth question, pick the wrong backbone. We have given certain trials and certain backbones when they were done in combination with daratumumab. So Pollux used Lendex as a backbone. Maya used Lendex as a backbone. Candor used Carfildex as a backbone. Griffin used Boti Lendex as a backbone. Castor used Botti Lendex as a backbone. Which of these is a wrong combination? Okay. Now, by the time the organizers, the event manager gets into the calculation of the top scorer, can we have the answers, please? Okay, the first one. Now, this happens to be PGIMER Chandigarh, where the first patient, next slide, please. Yeah. So the first paid patient was from PGIMER Chandigarh, but as part of the phase 4 trial, the first time it was used by Dr. Samir Melinkri. Now, this info was provided to us by J&J, so I don't take the responsibility for the accuracy of the answers. And in fact, they are the first one, the PGIMER Chandigarh, for using it in amyloidosis patients as well. Second question, please. Now, all of the following trials are related to daratumumab, except none of them is the right answer, because Pollex, Castor, Maya, and Cassiopeia, all 4 trials were discussed today in the Journal Club and the Journal Scan. And when Dr. Vincent was explaining the results of the Maya trial, so all of them are daratumumab based trials. So the correct answer is none of the above. Next slide, please. So the Pollex trial was a phase 3 randomized controlled trial for Dara RD versus RD in RRMM patients. Castor was phase 3 RCT, Dara VD versus VD in relapsed refractory myeloma patients. Maya was phase 3 randomized control trial, Dara RD versus RD, which was discussed today in transplant ineligible newly diagnosed patients. And Cassiopeia was about Dara VTD versus VTD in transplant eligible newly diagnosed multiple myeloma patients. So 2 were relapsed refractory and 2 were newly diagnosed. And out of these, Maya was transplant ineligible and Cassiopeia was about transplant eligible patients. Next question. All are labeled indication for daratumumab, except, again, next answer, please. Next slide. Yeah. So these are the labeled indications for newly diagnosed multiple myeloma and relapsed refractory multiple myeloma patients. And these are the different combinations and different things. So I will not waste too much of time because we are already running short of time. Next slide, please. Yeah, this is important to remember because when you give it in combination with Lendex, it's once weekly for 8 doses, followed by once in 2 weeks for 8 doses. Next slide, please. Now, there are different combinations of the backbones when you use in newly diagnosed multiple myeloma patients. And if you see, if you are using it in combination with VMP, it's once weekly for 6 doses, followed by once in 3 weeks for 16 doses. In Dara Lendex, it's 8 and 8, once weekly for 8 doses and once in 2 weeks for 8 doses. For Dara VTD, for the induction, that is pre-transplant, because this trial was used as pre-transplant and post-transplant. So in pre-transplant, for the first 8 doses is weekly, followed by once in 2 weeks for 4 doses. And for consolidation, following the autologous transplant, it's once in every 2 weeks for 4 doses. So the dosing differs depending upon the backbone that you people are using. Next slide, please. So this is the Dara Lendex, which was discussed today. So once week for 1 to 8 and followed by once in 2 weeks. Next slide, please. So the wrong backbone in the undermentioned trials is Castor. In fact, that was presented as a Journal Scan today, wherein Dr. Mona had clearly explained it was Dara VD versus VD. Next slide, please. So these are the different backbones. So we have already discussed about Pollex, Maya. For Candor, it was Carfilzomib. For Griffin, it was BRD. For Castor, it was VD. For Cassiopeia, it was VTD. For Alcyone, it was VMP. For Sirius trial, there was no backbone because it was a monotherapy which was evaluated. And for Equalus, it was Pomdex. Now, these are all IV combinations we are talking about. We are not talking about subcutaneous daratumumab trials. So these are all those where Dara was given as intravenous therapy. Yeah. So I think this is the last slide. I hand over. So before I hand it over to Dr. Joseph Jones sir for giving his concluding remarks, I wish all you people a very happy New Year and a Merry Christmas. And the next slide, please. And on behalf of Dr. Hari Menon sir and Dr. Pankaj Malhotra, the president of the Indian Myeloma Academic Group, we would like to invite you all to the upcoming annual conference at St. John's Medical College at Bangalore. Dr. Hari Menon sir had done exquisite, he had planned it very nicely and exquisite arrangements for making all you people comfortable. It's going to be a true academic feast. So we request all you people, whoever are participating, whoever are listening to us, to come and join us from 13 to 15 January at St. John's, Bangalore. With this, I hand over the stage to Dr. Joseph Jones, sir. And sir, if at all, if we have some minutes, we can, if at all, if any of the panelists have any comments or anything to discuss with Dr. Vincent, sir, so you can take it further. Over to Joseph Jones, sir.
Thank you, Dr. Uday. Thank you, Dr. Vincent Rajkumar, Dr. Abhinav, Dr. Arun, and Dr. Tilak. Wonderful discussion. Lot of queries. I think bringing in the epidemiologist was a novel thing which you have done, and that is greatly appreciated. And we will get more inputs on this. And thank you, Dr. Vincent, for taking us across the trials and alluding to the previous trials and giving us that insight into how the things work behind the scene. And it was great to see Abhinav's presentation, very clear and nicely done. And Dr. Tilak also gave a wonderful review on this and a lot of take-homes here. I wish we had more time to discuss with Dr. Arun on some of the nuances of epidemiology and how to interpret these later. And maybe we will take on that later. And I had put some questions in the chat box as well. Maybe later we can take it up. And I would request Dr. Uday and the participants, the organizers, to please show the results of the quiz for the prize distribution, and we have the two results to announce the winners.
Mr. Neerav, can we have the results, please? Okay, it's Dr. Aditya Jindal from PGI Chandigarh. So, Joseph, sir, you can.
Congratulations, Dr. Aditya Jindal, and congratulations to Dr. Mona, who has presented a wonderful presentation on the Castor trial as well. And over to Uday for the next meeting and announcing for the next participants here for the discussions.
Sure. So thank you, sir. Thanks for the closing remarks. And so the next meeting, we are going to have it on 26th of January, the Republic Day. So that's going to be the fourth Thursday. We are going to have it at the same time, 5:30 p.m. And the article details for the Journal Club and the Journal Scan, we are going to forward it to you in the next 3 days. And it will be coming to you as the mails and by the WhatsApp messages as we have been doing. And we will have one of our international fraternity members, most probably Vincent sir would be there to take up the moderation for the next Journal Club as well. And the article will be forwarded by sir to us in the coming 2 to 3 days. So we will discuss that. So with that, I think, thank you, Vincent sir, for helping us out in discussing this article threadbare, and also to give us an insight about how the trials are done and what are the nuances of the trials, what is behind the scenes, etc. So with this, I will just give the stage to Velu sir and Pankaj sir for any final comments and for closing this program for the day.
I'll just make it very short. Very nice to see you, Vincent. And I think that was brilliant, but you gave us an insight to the very obvious question which is asked repeatedly in all our fora. So I think you put it to rest. So you've buried many of these so-called doubts, which and how and why they are done by just asking one question: why was this trial done? So, and I must congratulate each of the, the presenters, the speakers, and the whole organization. Brilliantly done, and for Uday having put it together with his known perseverance. Thank you very much. I won't take any more time. Wishing everybody a very happy Christmas and a happy New Year. Jai Hind. Thank you. Over to Pankaj.
Yeah, no, I echo, sir, whatever you have said. So thank you so much, Vincent, sir, for giving an excellent oversight. So I think it has made my day, and I'm sure, it has made the day of all those people who are interested in, looking at reviewing the Journal Club. So thank you so much. Okay.
So good night, sir. With your permission, we'll close the session. So, Jai Hind, sir.
Thank you. Jai Hind.
Thank you very much.